This is Sciographies, an introduction to the people who make science happen. I'm your host, David Barclay. I'm an oceanographer with the Faculty of Science here at Dalhousie University. And on Sciographies, I interview different types of scientists about what shaped their interests, their career path, and how they get their research ideas. Thanks for joining us. In this episode, we talk to Dr. Aaron Newman. He's a cognitive neuroscientist, and his research aims to understand how different language experiences shape the brain. Dr. Newman also mentors STEM students interested in technology and innovation through his role as the director of the Surge Sandbox. Dr. Newman talks to us about how his lifelong interest in computers and coding led to a research career in brain imaging, his experience studying how the brain processes sign language, and his emphasis on innovative thinking when he's mentoring students and leading his research lab. I do like to begin somewhat at the beginning. So you're a Winnipeg boy?
Yeah. Yep. Born and raised in Winnipeg.
Okay. What part of the city did you grow up in?
I grew up in a neighborhood called River Heights.
Okay.
So sort of, I guess you could call it urban, but middle-class neighborhood.
Mm-hmm. The science bug, was that early on or?
Yeah, I think I came to the science bug. My grandfather on my mother's side was a medical doctor.
Okay.
And so that was always kind of in the family. I wouldn't quite say an expectation, but a hope maybe that people would follow in his footsteps.
Yeah.
And my parents taught at the community college on weekends, and I was over at my grandparents' place. And so every Saturday morning, I'd hear him on the phone, kind of making his rounds, calling patients.
So your parents taught at community college, but they also had other day jobs?
Yeah, so my dad was a school teacher. He taught grade 2, and my mom ran a daycare in our home.
Very interesting. So very caring, gentle people.
Yeah. It was very nurturing.
So in high school, would you say you would have sort of a variety of interests, or were you pretty focused on sort of the quantitative subjects?
Yeah, I was pretty scholarly in high school, and I was encouraged to focus on academics, and that was kind of the priority. So, I did track and field, but that was always secondary.
So university was maybe a bit of a given in your family?
Yeah.
Did you find it at all a difficult decision to make about when to go or where to go or if to go?
No, it really wasn't. I started at University of Manitoba, I think because it was the big university. People went there. My family had gone there.
Yeah.
And I just found it was very impersonal. The classes were very large.
Yeah.
I didn't, wasn't able to create a community or connect with other students that much. Very isolating. And I got, I guess, sort of last year of high school, first year of university, got very much interested in psychology and kind of, so in my family there was a history of addictions and mental health issues. So I started trying to understand that more and started understanding kind of the pharmacological basis of mental illness and that sort of thing. So that got me very interested in the field of psychology but also the neuroscience and the pharmacology and sort of the biological basis. And so when I compared what I could do at the University of Manitoba with Winnipeg, University of Winnipeg had smaller class sizes. They actually had more lab offerings in the psychology department. After my first year at Manitoba, I switched over to University of Winnipeg and was much happier.
Interesting.
I was one of those kids who wanted to go to med school, and even as I was interested in psychology and majoring, go in that direction, I was sort of thinking about clinical applications, neurology. I wasn't quite sure, but I was still on that med school track probably through second and third year.
So when did you take the turn and think more about research as a potential future career? Well,
I guess when I started at University of Winnipeg, I took the second year statistics class, and the professor in that class, Bill Simpson, I guess I did well in the class, and he approached me and asked if I wanted to be a research assistant. And so that, I think I started volunteering in the lab that year, and then that summer he employed me as a part-time research assistant, and that really got me going on, the research and just geeking out and writing computer programs. And he did visual perception research, and so it was very technical, if you step back, incredibly boring stuff, but it was kind of cool because you'd sit in a dark room and program these little displays with little dots of light and create optical illusions and depth illusions and that kind of stuff. So that really got me going.
So did you continue on working in that lab? That would be the end of second year, you said?
Yeah, I worked in that lab for a few summers and through the year, I think, as well, sort of volunteering. But, I liked the work, but I wasn't passionate about it. But the Canadian National Research Council opened up a research center right across the street from the University of Winnipeg, and they put an MRI scanner in there. And this was in the early 90s. So functional MRI for actually looking at activations in the brain, the first studies were published in 1992. This would have been 1996. So, very, very much early. On the cutting edge.
Yeah.
And this was, I think, possibly the second MRI scanner in Canada that was capable of doing fMRI. And one of the faculty members at the University of Winnipeg, Michael McIntyre, got an affiliate position with the National Research Council, and I convinced him to supervise my honors thesis. So I actually got to sort of break into that field and do my honors thesis with functional MRI. So that was a huge opportunity, and that really, set me on the course for the rest of my career.
Yeah. So can you describe to us, I think maybe we're familiar with an MRI. It's able to look inside at the brain's activity in real time. What makes it functional?
So the idea of functional MRI is that by tweaking what we call the pulse sequence, so how you acquire the images, you can actually get sensitivity to the ratio of oxygenated to deoxygenated hemoglobin. Okay. So hemoglobin carries oxygen through your blood, and when neurons become active, their metabolism increases, so they need more oxygen.
Okay.
And so more oxygen gets delivered, and it turns out there's actually a net increase in the amount of oxygen. So you get more oxygen than you need to those active brain areas.
Okay.
And so that's what the functional MRI scans show you, is that increase in oxygenated blood.
Looking for those oxygen molecules, really.
Yeah.
And then the idea would be that you're studying the activity of the brain by detecting where that extra oxygen is.
Exactly, yeah. Okay. So you manipulate, what the person in the scanner is seeing or what they're doing, and see which brain areas light up for language or visual perception or memory or whatever the task is that you're interested in.
So what was the task in your honors thesis that you were testing people on?
It was a language task, and we basically had people kind of free associating. I think we gave them one word, and then they had to generate other words associated with it. Okay. And some of the words were neutral, some of them were emotionally charged kind of words. And we were looking at what's the difference between sort of neutral language and emotionally charged language. Yeah.
So is there another part of the brain that gets activated when you say a word like hate or love or like that?
More areas get activated.
Yeah.
Now, it turned out in my thesis, they were still figuring out the physics of how this all worked. And so none of my data were actually valid. But, yet another honors thesis.
Yes. Yeah. You learned something. That's the important part. That's what I tell my students, This would have been relatively early on in computer world. So was computing a big interest of yours?
Yeah. Actually, that goes way back. So my great uncle, so my grandfather's brother, was in finance, and he would go over to Japan a couple of times a year, and he would always bring back like little electronic gadgets and toys and this kind of thing. Cool. And he really saw computers as the future. So this was, say, like very early 80s. He bought, so there was my mom, and she had two sisters, and each of them had two kids. So he bought a computer to be shared between the three families and sort of rotate month to month, because he felt it was so important, and he enrolled us in computer summer camp and this kind of thing. So it was like a very, very early stage kind of interest that got me going, and that just persisted with me throughout. So I think that's part of what appealed to me about that area of science, and maybe more than like the sort of wet lab animal kind of work. Was like, I like writing computer code and geeking out and playing with computers. So brain imaging is a great field to channel that. Yeah.
So from there, you went off to graduate school at the University of Oregon, and that must have been was that far afield for you? Did that seem exotic at the time?
It was quite the change. I grew up in Winnipeg. I'd lived there all my life. I'd done a bit of traveling, but not a lot.
Your experience there, was it mostly just right into academics? Was grad school like everything you expected it to be?
Yeah, it was pretty intense. But it was, it was nice because I really realized like you got past the undergrad just going to a bunch of classes and studying somewhat arbitrary things, and just everything you were doing was what you loved and what you wanted to do. But as it happened, my undergraduate supervisor had shared a house with my graduate supervisor.
Okay.
And so he knew her,
Yes.
and he had taught me a psycholinguistics course in my second year. And he remembered that I was really interested in the section on sign language in that course. And it fascinated me that here you have this language that is all communicated through the hands and through the eyes,
Yeah.
but it has all the same complexity, grammatical structures, and so on as a spoken language.
Sure.
And so Michael, my undergrad supervisor, told me that his colleague, Helen Neville, had just started doing the first neuroimaging studies of sign language. And he thought, I know you're interested in sign language. She's a great person. She's doing this work. You might be interested.
Yeah.
And so he wrote me a letter of recommendation. She phoned me up one day, and we started talking. And it was like, it was a very clear, like a good fit in terms of my interests.
Yeah.
She was very like enthusiastic and supportive.
Yeah.
And so it wasn't really a hard decision
Sure.
to go there. And it was very much like, go there. I'm going to do brain imaging of sign language.
Okay. When you're taking these images of the brain and looking at how the brain processes sign language or understands sign language versus spoken language, you're comparing the two, is that right?
Yeah.
So is it the same voice inside the head for someone who's using sign language versus spoken language?
Yeah, interesting how you phrase that question, because on the one hand, that early research and subsequent research has shown is that there really are a set of core brain areas in the left hemisphere of the brain that are specialized for language. And regardless of whether it's signed or spoken, those same areas get engaged and seem to do similar things. Like some areas are more involved in accessing meanings of words, some are more involved in stringing them together in sentences.
Yeah.
But as speakers and hearing people, we do hear a voice in our head.
Yes.
Right. And we sort of imagine speech that way. Whereas signers actually say that they imagine signing.
Sure.
Right. So, of course, especially if you've been born deaf, you've never heard anything, you're not going to hear. But it's a little hard to capture, but it seems like there is partly a perceptual part, but also partly just a sense of the motor part, like the actual activity of generating signs.
Right.
As they're imagining.
Yeah. Very
sign
interesting.
language,
So it is slightly different. Very,
exactly.
Very cool.
Yeah. And then our research also showed that there were, in addition to the same core language areas, there were actually some areas in the other side of the brain, the right hemisphere, that seemed to be uniquely involved in sign language and possibly in processing the more spatial aspects of sign language. Because there are aspects of the language that involve knowing like who's in what location in space, and that determines grammatical relationships and so on. So we don't really have that spatial component in spoken language.
You asked these questions about language. That was the basis of your master's thesis?
Yeah, so I was very, very lucky in that I went to grad school in Oregon, and we didn't actually have an MRI scanner at the University of Oregon.
Okay.
And my supervisor was collaborating with folks at the National Institutes of Health in Maryland, so on the East Coast, to use their scanner. And that worked out well because Gallaudet University is the only deaf university in the United States, and it's based in Washington, D.C.
Yeah.
And so it was a good place to recruit participants and have them come in. And so within the first few months of going to grad school, I was actually sent out to Washington to help collect data from deaf people. And the study was comparing people who were born deaf and learned sign language from their parents to people who were born hearing to deaf parents, and so learned sign language as their native language, but had normal hearing. And then the group that I was involved in acquiring data from were people who had learned sign language later in life. So they just become interested in being like a sign language interpreter. So they've gone to school and become very fluent. So there's in the psychology of language, there's something called the critical period hypothesis.
Right.
And so the idea is that if you're exposed to a language early in life, you learn it quite fluently. But after a certain age, it becomes much harder. And so we were interested in that, but in sign language. Right.
So did you pick up sign language then?
Very slowly, yeah. I took a sign language. I guess I took 2 sign language courses when I was in grad school.
Yeah.
And I could stumble through like introducing myself and being polite, but
say
not
like please hold your head still in the machine.
Yeah, exactly.
I'm good at saying that. Yeah. So this must come down to the kind of idea of neuroplasticity and how that changes over time.
Exactly.
Yeah. And so we're less neuroplastic as we get older.
Yeah, that's the idea.
So did you decide to stay and do a PhD kind of as soon as you started, or?
The program that I was admitted to was sort of a PhD track. So it was kind of expected when you went in that you would get the master's along the way, but you would do the PhD as well. So I was pretty much, mentally committed from the time I arrived there to doing that.
Yeah. And can you sort of just give us the summary of your PhD work?
The question was around what's different between native speakers and late learners of language, and are there differences between different second languages? But what was interesting coming out of that was what we found was that so there's this notion that if you learn a language later in life, you're not as good at it. And then the question becomes like, what's different in the brain about that? What we found was that if we looked at language proficiency as a continuum, both in the native English speakers and the Spanish speakers, the way that brain activity was modulated by proficiency was the same in both languages. We all know people who are incredibly fluent in English and other people who get by, but have a smaller vocabulary, less varied language and that sort of thing. And so it does seem to be much more of a continuum and not just this sort of hard stop that there's something different about the second language. You had a brief stopover as a postdoc before coming to Dal. Yep. Can you tell me a little bit about that transition? So I did my postdoc at the University of Rochester in New York State. And I mentioned before Gallaudet University in Washington, D.C. It's a deaf university in the U.S. And then in Rochester, there's the National Technical Institute for the Deaf, which is the only technical college for deaf people in the U.S. So again, we had a very large population of deaf people in Rochester and sort of young college-age students who were eager to participate in brain imaging experiments. So that's where I did, the bulk of my research there was on sign language. And it was a great community. We had Ted Sapala, who was one of my supervisors, who was deaf himself, deaf postdocs, deaf faculty members. So it was a rich environment. My sign language got a lot better then. It's gone downhill since then. But it was great to sort of be in that environment and really see sign language is a living language and get to work with people and do research in that area. And it was also a unique time because, again, I have this tradition of like going to places that don't have MRI scanners in order to do MRI research. But while I was a postdoc, we did get a new scanner and opened a brain imaging center. And I was actually put in position in that center where my job was basically to onboard new researchers. So largely people coming from the medical school who had ideas for MRI projects, but they had never done fMRI research. And so I would sort of consult with them and help them design an experiment and get it going. So that was a lot of fun because that's sort of the very creative aspect of science that I like. It's like experimental design and just brainstorming and, learning about a new topic and thinking, how can we apply this to MRI? And then not having to follow through on analyzing the data and writing up the paper.
Someone else can do that.
Yeah, exactly.
So that must have set you up pretty well to come here to Dal and become a faculty member.
Yeah.
And you're cross-appointed to these
so
other schools now.
I'm cross-appointed in psychiatry, surgery, and pediatrics.
Right, which is quite a broad, almost going back to when you started, quite a broad area of interest, right? So how did you kind of come across this idea of trying to make new techniques, new technologies? When
I came to Dal, first of all, I got involved in a number of research projects that were very clinically focused. And so that started getting my mind towards, how do we actually take, in my case, neuroimaging and answer questions around like cochlear implant outcomes or stroke patients going through rehab.
Right, applied to real patients.
Right, yeah. So that got me thinking about the clinical applications. And then we had the opportunity to apply for an Insert Create grant, which is a training grant. And so then we had funding to create Radiant, a neurotechnology graduate training program.
Obviously, when we think about neuroscience and the brain and technology, there's lots of tasty kind of dystopian products like reading the minds of consumers or whatnot. But what have you found most students have come up with when they're sort of thinking about, commercializable products in your field? There's a lot of variety, but I would say that the vast majority of students that I've worked with are very mission driven. Like, they're doing this because they want to help people. Right. And they want to develop clinical applications, medical applications, whether that's for PTSD, stroke, concussion,
those kinds of applications. That's usually where you see the interests. Yeah, it's not so much in neuromarketing and mind hacking.
That's I'm glad to hear it. What are sort of the most exciting kind of innovations you see happening in the field?
Yeah, it's an interesting question. The technologies we have for brain imaging are all kind of crappy.
Yeah.
And you can quote me on that. Like, on the one hand, they're super amazing, right?
Yeah.
Like, functional
Absolutely,
MRI, we can see down to now sub-millimeter level of resolution and brain activation, and on the, sub-second level. But you need this huge multi-million-dollar machine with a huge magnetic field around it. So it's not very practical as kind of a day-to-day tool for doing anything besides understanding the brain. And then we have more portable technologies like EEG that are, have a lot more limitations in terms of what they can tell us. And to get good signals, you have to use gel under each electrode, and so your hair gets all goopy and this sort of thing. So there isn't really sort of a robust, just like put a hat on your head and go decode your brain activity kind of thing. But I think there is a lot of opportunity for like neurofeedback, so helping people, decoding brain activity and feeding that back to people to help them engage in more productive or beneficial areas. So, for example, one of the companies that actually did come out of our training program is called Axum Neurotechnology, and they're based here in Halifax, and they're working on developing a near-infrared spectroscopy imaging system for stroke rehab. And so the idea is that if you've had a stroke that impairs your ability to move your limbs, then one of the ways that you can actually encourage the brain to rewire and recover that capacity to move your limbs is just to imagine the movements and re-engage those neurons.
Amazing.
But, to tell somebody, just sit there in your bed and imagine moving your arm for 15 minutes every hour, like that gets old fast. But the idea of neurofeedback is that if you have a brain imaging system that's measuring the activity in the brain, as you imagine those movements, you can turn that brain activity into some sort of visual stimulus, gamify it, and give people some feedback that encourages them to keep doing it and kind of know when they're doing the right thing, when they're not doing the right thing. And that sort of thing. So, I think there's a lot of opportunity there.
Right. You mentioned this type of technology is very beneficial for people like stroke patients trying to recover and helping them recover. Is that kind of the primary motivators for most of the technologies you see is on the aspect of, picking a particular neurological condition and trying to understand how we can, improve the outcomes?
I think there's certainly a lot of work that is clinically focused, but there's also a lot of basic research that's still going on.
Okay. So in terms of neuroscience and sort of the progress in neuroscience, where do you think we are if you compare it to, say, like classical mechanics? Like, are we at the point where the apple is falling on Newton's head, or are we looking at quantum gravity?
Yeah, I think we're maybe a little past the apple, but not that far past the apple. Yeah. The brain is incredibly complicated. I don't think anybody has any questions about that. But, a single neuron could be connected to 10,000 other neurons. And those connections aren't stable. They change on the time scale of, seconds and minutes and hours and days.
Yeah.
And so, trying to capture that and understand that, a single neuron, and there's billions of them in the brain, right? Yeah. And when I say that all the neuroimaging tools we have are crappy, right? Like, so if you do fMRI, we have a spatial resolution, we could push it down below a millimeter. Still, in like a cubic millimeter of brain tissue, you've got thousands, tens of thousands, hundreds of thousands of neurons, all interconnected, lots of different types of neurons, and then supporting cells like glia that also play functional roles.
Right.
And so there've been some multi-billion-dollar efforts to computationally model networks of neurons. And the scale of that is still tiny compared to the brain. And so I think in terms of our understanding, just that level of structural complexity, how things work together, and many different neurochemical systems, there's so much to unpack that we're barely scratching the surface. It's amazing we can do anything, quite frankly, but we're there's a lot of job security in neuroscience, I'll tell you that.
So from running the Create Radiant program, that kind of spun off into another project where you're running a sort of a sandbox for innovation?
Yeah. So the Create program, we had funding for 7 years, and it was non-renewable funding. Mm-hmm. So I think it was a great experience, but it had to end. Mm-hmm. But then as it happened, the Faculty of Science at Dal got funding from the province of Nova Scotia for a science sandbox. And the basic idea was to foster innovation and help create more of a knowledge-based economy in the province and help us, diversify our economy and do more interesting, innovative things with science. So that's been a great opportunity to explore different ways of training students and really open students' eyes to what they can do with science.
This is the kind of training that most students don't normally get in their regular classes, right?
Yeah, exactly. Yeah, it's how do you actually take what you're learning in class and apply it to real world problems.
Yeah, can you tell us a little bit about some of the activities in the sandbox and some of the success stories that have come out of it?
Yeah, we've tried to do a lot of different things. Like even the name Sandbox is suggestive, I guess, right? So it's an opportunity to play and try things, low stakes. So we've done hackathons, which are big in computer science, where people would come and be given a challenge and write computer code for it over the course of a weekend. We don't do coding hackathons because a lot of science students don't have that kind of background. But we've done hackathons around brain-computer interfaces with EEG, so that's something I'm familiar with. Yeah. We did a VR applications in science hackathon. We've done Ocean Startup Weekend challenge, where people come and just brainstorm ideas around, broad challenges in the ocean space. Last year we did a ROV hackathon, so remote operated vehicles for underwater exploration. So the idea with those is just give students the opportunity to come and get their hands on a technology. Yeah. And not necessarily worry about breaking it or is this on the test? Sure. How do I get an A? It's just what can we do with it? And play with it and just imagine. And it's really empowering. It's really exciting to run those events and see students just grasp on and bring in knowledge that they've, from, social media or popular culture or whatever, and imagine the possibilities and play away with it. And it also brings together people from different backgrounds. So we advertise broadly. So with our neuro hackathon, we had teams where you've got, first year computer science student, second year psychology student, master's student, mechanical engineering. So these are three people who would never encounter each other on campus in a typical day. But they're all coming together and bringing their unique experiences and talents and ideas together on this problem. And that's the magic. That's what really drives us. Cool. So we do those kinds of events. We run a class that I teach on science innovation, which is a full year. And I really wish more science students would take it. Because I think it's a great opportunity, and the feedback I get from it is great, because we really walk people through this process of human-centered design. How do you find problems, talk to people, understand problems from their perspective, and design solutions that are actually appropriate to those kinds of problems? And then take it a little further and think about how then could you make that solution viable and scalable. So thinking about what we call business models, which is not necessarily how do you make money off of it, but any innovation is going to take a significant amount of human resources and financial resources and materials just to test it, develop it, bring it to market, ensure that it's safe, passes all the regulatory kinds of checks and all that kind of stuff. So it sort of goes into entrepreneurship, but we have to be cautious because it's not the sort of heavy capitalist entrepreneurship. It's more this is how we package things to actually bring a solution to reality.
Thank you very much for coming in and talking today. It's been fascinating.
Yeah, thanks for having me. It's been fun. Great talking to you.
In our next episode, we'll talk to Dr. Erin Bertrand. She's a marine biogeochemist looking at the relationship between microbes and ocean metabolism. If you are enjoying Sciographies, hit subscribe and consider rating our show. I'm your host, David Barkley. Thanks for listening. Sciographies is brought to you by Dalhousie University's Faculty of Science and CKDU 88.1 FM in Halifax, Nova Scotia. Our producer is Nicole Killowy. You can learn more about Sciographies at dal.ca/Sciographies.